Hello, hello, and welcome. Hello, Johannes. How are you?
Hello, nice to see you. Nice to hear you.
Nice to see you.
I'm
Nice
ha
to hear
I'm
you too.
yeah, I'm happy now to discuss on fasting and the brain and how lipids are activating brain circuits. Yeah.
Oh, we're so excited. We're so excited.
I hope everything works. I send two PDFs, one on the paper and one on the model we are working on in
Okay.
more detail. I hope
Yeah.
you could load it to the audience.
Yeah. No, there's an option to put a link, so we're gonna do that. And that's why we start the room, a few minutes before. So we're just setting that up to
Perfect.
make sure. Yeah. So don't, you don't worry about that. We'll take care of it. I
Okay,
think
because
Katerina will be coming in. Yeah.
yeah, last time I had some problems with the technique. I couldn't speak. It was really awful. Yeah, but I hope everything is fine for today.
Oh, yeah. I hope so too.
Yeah, but
Hi,
we have an
Jamie.
excellent audience. Hello.
Yeah, hi. Hi, Jamie. How are you?
Hello, Doctor. It's a pleasure to meet you. I'm Jamie. Hi, Cecilia.
Hi, Jamie.
I'm looking forward to your talk. I was just reading your paper. It's incredibly interesting. Um,
I'm
I,
happy
I
to
was
hear that.
I was about to ask you some questions, but no, that can wait. That can
Sure,
wait.
sure.
Uh,
I'm.
yeah.
It's really a new field of research, and I also have a lot of questions. Yeah, still,
Good. I
which
love to hear that.
we
Yeah.
couldn't answer everything in one paper. I mean, that's yeah, it's too much at one time, but it's interesting. So how it is, yeah.
Well, it would actually be worth having a paper written on someone answering all the questions in one paper. But that would be a feat, yeah, if we could answer all the questions in one paper.
Yeah, yeah, it's always like this. Yeah, I
But
mean, either you go into deep with some single questions or you are very broad and then you are not able to follow them into deep. Yeah, that's always the point. Yeah,
do
but
you ever?
yeah,
Sorry, please continue.
now every anyone has this problem. Yeah, I mean, sure.
Mhm. Do you ever feel overwhelmed by for every question you get an answer to, two or three more pop up? Does it ever feel overwhelming?
No, that's working. I mean, we go step by step and we try to tackle a problem from different ways and with different animal models, and we try to better and better understand the system. I mean, it's yeah, I don't know if you call it overwhelming, but it's yeah, it's complex. Yeah,
Mhm.
you have sometimes to step back and rethink your strategy, but yeah. So now we have even a big loop where you start at the hypothalamus, you go on the lipolysis, and then this lipolysis brings in some lipids which go into the brain, and only you have their specific transporters, and they are in the brain. They make a very complex signaling at the synapse, which we have described over time. So it's not a one paper shot. I mean, we started 2009 with a Cell paper where we first described that lipids are able to modify signaling and synaptic glutamatergic release. And then, yeah, so we didn't know what to do with this, to be honest. Yeah. It was, these proteins are very unique and you don't find any homology. So you're completely a little bit on your own. Yeah. But took 12 years. Yeah. To understand. Yeah.
Wow, 12 years. So
Yeah,
much
but in
time.
this time, I mean, it's like a salami tactic. Yeah, I mean, you start with one problem, go to the next one, go to the next one, and then I mean, what we thought at the beginning, it's a psychiatric molecule. Yeah, so we have schizophrenia and things like this. Yeah, and then we found some humans which have a characteristic mutation in these genes, and they did not have a schizophrenia, but somehow slightly increased risk. So it was not what psychiatrists would say this is a single gene responsible for schizophrenia. No, it's possibly one part of the system, but one part also which has much more to do with fasting, and it may be something which is an ancient circuit which keeps us alive when we are missing food. Yeah, I mean, you always have to think. I mean, it's very understandable if you say when you are fasting, you don't have food, you get more active and you are searching food. And I mean, humans have some quite of reserves, so they can stay some weeks without food if they have enough water. But mice are at the limit mostly, yeah. And therefore, it's worth to study this in mice because what happens in humans is happens much faster in mice, and so we are able to describe these changes even after 16 and 18 hours of fasting in mice. Yeah, and then it seems somehow logic, yeah, because if mice do not have food, they have to search, and it's not like we keep our mice in a very pleasant conditions. They have always food, they have nice embedding, they have social interaction with some wheel where they can running wheels. Yeah? In nature, it's always food is limited, and you have to have some mechanism which keeps the animal alert and searching for food. And that's very difficult to understand if you're going straight forward. Mostly, I think that results come if you come from the side, yeah, from the sideline, yeah, and you say, oh, somehow my gene has to do something with food intake, and then we followed, and indeed we found something.
That is incredibly interesting. Yeah, I'm actually looking forward to this talk because I always find it incredible when we can look at mice and actually find so many correlations with like ourselves. Like obviously, like biologically, there's a lot of things we don't have similar, but what you're saying how learning about things like fasting with mice and everything, and that actually can translate over to humans. I always find that amazing when I find out how similar we are to different creatures. It's always amazing to me.
Yeah, I mean it's a wonder on one side. On the other side, I think this mechanism are related to the nature, and they develop in a similar way in in different species. I mean, we are 70 million years away from the mice, and yes, that's the point. Yeah, and I think we are, yeah, some even more different from pigs, and still you may think that the mice have a lissencephalic brain, so they have a non-folded brain, while we have a folded brain. And pigs, which are quite more different, have also a folded brain, meaning indeed this mechanism are simply the best way to cope with limited space or with limited food, and they develop in parallel. And yeah, possibly that's one of the clues why we are able to study in different species similar things that then apply to humans. Yeah.
Yeah, I mean, that's all really, really interesting. I mean, I can't wait to hear this talk. And, it's actually, 2 o'clock now. So I think we should try and see, how we can get started. Unfortunately, Katerina is trying to get in, but her internet is not very strong where she is.
Okay.
So yeah, unfortunately, I don't know, but she might join us soon. Hopefully she joins us soon. So yeah, but just to confirm before I go into, introductions, your last name is pronounced Vogt, right?
Yeah, it's Vogt. Yeah, Vogt. Yeah,
Vogt.
Vogt.
Oh,
Yes.
I'm so sorry. Vogt. Okay.
Yeah, but it's difficult. I'm sorry.
No, it's okay. Thank you. Vogt. Okay, I got it now. All right. So yeah, I guess we're just gonna get started, and hopefully Katerina can join us soon. So I will give a brief introduction of you, Johannes, and then Jamie, could go ahead and ask you a couple questions. Just so we can get to know you a little bit more. Okay. So. Right.
Perfect.
Yeah, welcome everybody to the Science Society. Today we have Dr. Johannes Vogt, and he'll be talking about brain signal pathways, controlling food intake. So Dr. Vogt, he is a principal investigator working in the Department of Molecular and Translational Neurosciences in the Center of Anatomy in University Hospital of Cologne. And his group works on synaptic lipid signaling, which regulates glutamatergic transmission and thereby cortical network excitability. And so the research is focused on metabolic changes that affect bioactive lipid levels and thereby also synaptic lipid signaling, which lead to cortical hyper excitability, and a typical finding in aging brains. So he's interested in elucidating the mutual connection of synaptic lipid signaling, metabolic regulation, aging, and cognitive functions. So he and his group have identified a group of molecules regulating synaptic lipid signaling called plasticity regulating genes, which regulate glutamatergic transmission in in the cortex and thereby cortical network activity. So he's going to have a wonderful talk with us here about just how brain activity works and what occurs when, we eat food. So I have posted up top the link to the article, his article. It's an Adobe link. So please go ahead and click that and follow along as he gives his talk. So I'm going to pass it on to Jamie. Hey, Jamie. So Jamie's going to ask you a couple questions just so we get to know you a little bit more and then we can get into the talk.
So thank you very much. First of all, thank you for this wonderful introduction. So I'm happy to take questions.
Thank you very much. And so, first of all, let me officially welcome you to Science Society. It's a genuine pleasure to have you here, Doctor. So, first of all, just to give the audience a way to just get to know you a little bit better and make it all friendly. The first question to you is, so when was the first time you actually felt yourself having an affinity for science and academia? And is there any story behind that? That's my first
Yeah,
question.
so that's quite a long story. So I'm trained as a neurologist. So I'm a medical doctor trained as a neurologist. And during my PhD, I worked on a research topic and yeah, some kind related to stroke and some potassium channels. And I was always fascinated by that. And in fact, during my medical education, I was fascinated by neuroanatomy, and I thought it's so wonderful. I mean, you have so much wiring on so little space, and everything goes so smooth, and it's so precisely organized. How can that really happen in nature? Yeah, and I think for me that was a miracle especially the fact that I mean, whenever you see a brain, you start and look at brain and you are fascinated and you think, oh, the brain is so complicated. You have so many wirings and so many gyri and sulci, and they have all names and all they have some kind of meaning, yeah, and you have to also to irrigate this, and you have the blood supply and the venous regulation. And then the interesting thing in the brain is you think it's so complicated when you have an outside look, and then you go into deep and you see, oh my gosh, that's even more complicated. And in really tiny spaces and tiny slides, you find so much detail, and the brain is so much full packed. And then you see a lot of neurons, and you see a lot of spines, and you go further and see, yeah, you go with electron microscopy and you think, oh, now I discovered some world. Yeah, and then you start doing biochemistry, and then you see, my gosh, that's even more complicated. So there are so many proteins there. One synapse, you don't see it by eyes, you barely see it with a microscope. And then you go to electron microscope where you are able to visit it and to have a look on it. And it's even more complicated because you have so many proteins and all do react with each other, and everything goes so fast. And this is really, if you shut that down, you shut thinking down and being down. And how can that really happen? I mean, it's really something like a miracle. And therefore, I was fascinated by that, and I said, okay, I would like to go forward with neuroanatomy. And yeah, so we happened to be then engaged with this protein PRG1, which we found in a screen, which was, in fact, to be honest, we did not expect. We did neuronal regeneration. So we had lesioned neurons, and then we wanted to see. So the theory is that a lesion neuron is trying to regenerate itself, and if we find the proteins which are upregulated trying to bring this regeneration forward, we will be able to enhance this regeneration by overexpressing this protein. And now we found this PRG1, which was completely new and had no similarity to any kind of other proteins, but some kind of similarity to lipid phosphate phosphatases, something I never heard before. Yeah, so okay. We said no homology, no clear motifs, yeah, but transmembrane protein seems somehow nice, and with its homology to lipid phosphate phosphatases, it has something to do with lipids. Now we said, okay, let's start trying to find something out. Yeah, so maybe this is a, I don't know, you have a housekeeping protein and looks quite nice, but you don't know where it's expressed, possibly everywhere. If you knock it out, nothing happens. So that can happen. Yeah, I mean that's quite something that happens. And then we made a knockout mouse and made an antibody again. So that was really a big step forward, because we were able then to show the antibodies we produce are also specific, because we had very big problems with unspecificity. So trying to correlate with a knockout animal to see how specific is the antibody, which one to take. And then we found that this protein is expressed very specifically in excitatory neurons in the brain only. Yeah, so that was already something new, because normally you have the proteins almost everywhere. Yeah, and we have only in excitatory neurons, in the principal neurons. And that was already nice, but then we went further and did indeed what I talked before, electron microscopy. And so, wow, this protein is only expressed in excitatory synapses. So not the inhibitory synapse, only the excitatory synapse, is not expressed in astrocytes, is not expressed in oligodendrocytes, is not expressed in microglia. So very specific excitatory synapse on excitatory neurons. So I thought, okay, that's nice. And we only found it at the postsynaptic density, where where you have all the receptors for glutamate. Yeah, so the AMPA receptor, the NMDA receptors. These are the ionotropic receptors, and you have metabotropic receptors. You have kainate receptors, but they seem to be this PRG1, this plasticity-related genes, seem to be there specific for. Yeah, and I will only talk about PRG1, yeah, which is possibly the most known member, the best known member of this family, but not the only one. But it's well characterized. And
Well,
yeah,
oh,
Jamie.
sorry, I was just going to say that your fascination for this is really, really interesting. Your interest in this is very contagious. I'm actually getting quite excited as you're even describing this much. And last question then before we put you onto the talk itself.
Sure.
Can you please tell us then what was it that led you? Well, I mean, you were actually already explaining that, so I apologise for interrupting if I did. But
No
what was
problem.
it
Sorry,
specific?
I was talk too long. Too long. Yeah.
Oh, no, 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 no. That's why you're here. That's why you're here. Please, please, no, no. The question was just if you, what was it that got you studying this particular aspect of this paper that we're going to talk about today?
Yeah.
That's my
So
first question.
in fact, we found a lot of things on this plasticity-related genes and synaptic lipid signaling, and this was very related to what happens if we delete PRG1. Yeah, and we thought, yes, we delete PRG1, we have more lipids in the synapse. And we always thought, how can it be? I mean, you have to have a bigger context on that. Yeah, I mean, it's clear if you delete PRG1, you have more lipids. These lipids are bioactive, but you have to have, I mean, most research on lipids. It's in the periphery. I mean, you have the adipose tissue, you have the brown adipose tissue, the white adipose tissue, you have the liver, you have all this diseases which are metabolic disease and are affecting the liver. They affect the pancreas. It's a lot of disease you have. And then we thought, we have to have a link. I mean, it cannot be that we have a lipid, we have a really very precise lipid signaling, and we don't have the big view. I mean, it's like when you are a little bit of looking at something very close with a microscope, and you see some kind of very interesting findings, but you do not realize that you are in the jungle. I mean, that is, you see the really things you analyze under the microscope, but you are not able to see the jungle. And this is what the feeling we had, and we said, it cannot be. We have to have a bigger look on it. And then we said, okay, we analyze what happens if we fast animals. We started to make research, and what everything one would believe, then you have a lipolysis, and when the glycogen storage are depleted, and yes, sure, but lipids are not able to pass the blood-brain barrier. Then we got in contact to some colleagues which are analyzing this mechanism, which are able to transport lipids past the blood-brain barrier. And yes, then we started to said, how can we challenge the system, because we want to have something which is very specific, and it's not, and it's really related to lipid changes in the body. Yeah, and then we come on fasting, and we tried different fasting protocols and so forth. So this is the story how we started, then go step by step until we teamed up with people who are working in the hypothalamus, and to our big surprise, they show indeed this agouti-related proteins. They are affecting peripheral lipolysis. So meaning there are neurons in the brain. Really, you have to imagine neurons in the brain, which are able to affect the fat consumption and the lipolysis in the body. I mean, that is really crazy. I thought, wow, we have to learn more about this. And so we teamed up and did our experiments, and we saw, oh yes, indeed, that makes sense. That makes sense.
That is incredible. What a wonderful lead up to your talk. Okay, so without further ado, then, Doctor, thank you very much for sharing that with us. And when you're ready, the stage is yours, and we're looking forward to hearing your talk. Thank you.
Thank you, Jamie. No, you possibly we do it like always. So I talk about a topic. I did not prepare now a talk like you would expect to say a talk. I mean, I thought we go through the paper and analyze possibly. We discuss the caveats and what we found, and yeah, possibly it's best to start with the synapse. I mean, I think anyone is familiar with the synapse, and we have in fact in the brain basically two synapses, two classes of synapses. And the one are inhibitory synapses, which are bringing a neuron, which are inhibiting a neuron, and the other ones are excitatory synapses, which induce an action potential and go the way to the excitation of a circuit. And we found that this molecule is indeed expressed very specifically, as I said before, in excitatory neurons. So these are the neurons, about 80% of the brain neurons, which confer excitation in the brain. And it's only expressed in excitatory synapses at the postsynaptic density. And this is something you have to think about. The synapse is like a sacred place. So if you have a presynaptic terminal where you have all these glutamatergic vesicles, and when you have enough calcium in the presynaptic terminal, you have a release of glutamate, and glutamate is activating the receptors at the postsynaptic density, mainly AMPA receptors. If you have enough glutamate, you are affecting also opening the NMDA receptors. And you have then an entrance of positive ions, yeah, mainly sodium and calcium. And this leads to a depolarization of the neurons and to an action potential, and this makes the neural circuit work. Now, this lipids, this PLG1, which is located there, in fact, it has nothing basically to do with excitation. It's simply, it fishes out LPA from the synaptic cleft. And yeah, so in fact, this is a negative modulator. Yeah, so we thought, okay, a negative modulator which affects a presynaptic molecule. I mean, a presynaptic release. This is not so easy to understand because this pre and postsynaptic terminals, when people analyze them, they basically are not analyzing both at the same site. So you have a lot of people which are analyzing the presynaptic terminals. They analyze how these glutamate vesicles are able to release glutamate, and there are tons of paper and very, very clever people working on it on the labs. And there are the other people working on the AMPA receptors, the same, almost same amount, and very clever people. But we are looking now for something which is connecting the presynaptic release with the postsynaptic excitation. And indeed, to connect this, we found that there is a LPA2 receptor. And I hope that you have a view on this PDF, Adobe PDF, which I sent. There you see the presynaptic LPA2 receptor is a G protein coupled receptor, and one of the very few which is increasing via IP3 the calcium in the presynaptic terminal, and therefore it's modulating the glutamate release. So the more calcium you have, the higher probability to release glutamate and to induce a postsynaptic current in the neuron, which has the receptor and which is innervated by this presynaptic terminal. Now, so far so good. The question which we had is, where does this LPA come from? And LPA means lysophosphatic acid. Is a very small lipid, one of the simplest lipids, but it's a very potent activator. And it's very rapidly degraded. So it has a very short half-life time. So it's in the blood, you have it in minutes. So within 1 minute, you have if you block the production, you have a decrease of LPA. So it's something which is very strictly regulated. So it cannot come from far away in the brain. It has to be produced somehow in place. And we thought the most closest relatives, or the most closest structure, are the astrocytes, which in the excitatory neurons, they are activated, they are insheathing the synapse. And you see, this is now at the number 1. Yeah, so we started backwards. We started with number 3, the PRG1. Then we went to number 2, the LPA2, and then we came to autotaxin. And in fact, I started with PRG1 because this is the logical way to start. This is how we found everything and how everything makes sense. When we tell the story, we tell it mostly the other way around because it's, yeah, possibly it's nicer to show. But in fact, it's really the thing we are starting with the bottleneck. And this is indeed PRG1. And then we come also to autotaxin. This is number 1. And we found that astrocytes, they express autotaxin. And you know, astrocytes, they are cells which are enormously ramificated, and they have contact also to inhibitory neurons. They have contact to blood, to the blood-brain barrier. They have contact to other neurons. So they are have very many contacts. But autotaxin was only specifically expressed in excitatory synapses. So we thought, wow, that's somehow interesting. Yeah. So we never found autotaxin in inhibitory synapses. And what we know from the work of colleagues is that autotaxin is producing lysophosphatidic acid, this is LPA, and it's producing from LPC, and this is lysophosphatidyl-choline. And by a so-called nucleophilic attack, there is a choline group which is removed, and then you come to LPA, which is the bioactive part of this. And the beauty of this is everything happens at the synapse. But sure, this is not so easy as it seems, because autotaxin is packed in vesicles somehow, and autotaxin is something like a carrier also for LPA, and autotaxin forms a tunnel, and it has an integrin binding site where it can bind close to the receptor. So in fact, autotaxin is shedded from the astrocytes. It's traveling within the synaptic cleft. It's producing LPA. It's keeping LPA in the tunnel, and it's binding close to the effector site via an integrin binding site. So this is quite a complicated story in a very small room, yeah, which is the synaptic cleft. And here we are talking about 20 to 30 nanometers. So we are far below the light microscopy, and we are at the edge of what normal stats are able to show at the moment. So something about 20 nanometers. So sure, there are also better machines. The Miniflux from Iberia, they are able to have a resolution of 3 nanometer where you see single proteins, but these are very expensive machines, and I know only very few people which have such a very expensive machine. Okay, possibly before I start, are there questions to this mechanism, which we had some trouble to find how it works?
At the moment, I don't have any. I'm just trying to absorb everything. Sister Reem, do you have anything at the moment?
No, I'm good. I was just enjoying that wonderful background. Please keep going.
Yeah,
Oh, if
please,
so is
please.
Katarina. I don't know. Katarina just came in, so I don't know. Maybe she has something.
Okay, good. If just interrupt me, please. I'm just we can just then talk about the next things, yeah, and or come back, yeah.
Absolutely, thank you.
Now, now we said, okay, we know that animals, when they fast, they have around 12 hours, they have a reserve of glycogen, and then glycogen storage are depleted. We know this from analysis in the liver. So therefore, we said, okay, we go beyond this phase where glycogen storage are still available, and we go in a fasting period where we have also lipolysis. And we said, okay, 18 hours is something we are targeting for, yeah, because then we are in the middle of the lipolysis phase. And what we did, we took plasma from these animals, and we saw, indeed, if we look for total plasma, we have an increase of these lipids, this lysophosphatidic acid, in the fasted animals when comparing to the control animals. This is figure 1b. And it's not only the total plasma, but there are some specific LPA subtypes. And by specificity of LPA subtypes, meaning this is the chain length. So the more carbon atoms you have on the backbone, so the bigger the chain. And if they are non-saturated, they have no double bonds, and each double bond is shown by the second number, and it shows the degree of non-saturation, so how many double bonds it has. So these are non-saturated lipids, and you see they are almost all increased. And we looked for 18.1. Why 18.1? 18.1 is the most known lysophosphatidic acid. It's very potent in the cardiovascular system. It's in fact the most potent. It's we have it in quite amount in the blood, and whenever you look, people always work with 18.1 because they have the best results. And we said, okay, why not looking for 18.1? We are looking for 18.1, and we are measuring also in the brain. And in the brain, it's difficult to measure in the synaptic cleft. So that is very simple. Yeah. So I think even in 20 years from now on, we will not be able to measure LPA or any other things in the synaptic cleft because this is a very small region, which is about 20 nanometers in one direction and something about 300 nanometers in the other direction. So we only can do some dialysis, but in fact, we cannot measure LPA there. But we can measure in the liquor and in the CSF. We have comparable conditions like in the synapse. So the CSF is something like a proxy for what happens at the synapse. It's not exactly a proxy, but I think it's the closest way we are able to go to. And indeed, we found also in the CSF that LPA81 is increased. But when we look at the other species, we didn't find the same increase like in the blood plasma. We have only specific increase. We have 18, 16, 0, 18, 0 increase, but not 18, 2 and 20, 20, 0. So not the longer ones. So it seems to be quite a specific change. Now we wanted to see what happens with the brain when we fast, and we did miniature frequencies. And I don't know if you're familiar with electrophysiology. When you do miniature frequencies, in fact, you measure the release of some kind of single vesicles. We call them quanta. So it's really, we have a single, yeah, this is a proxy for a few vesicle release. So it's what happens when you have no input in the axon terminal, but only the spontaneous release. So we give therefore tetrodotoxin, which blocks all fast sodium channels, and we have basically no activity. We have only the fusion, the spontaneous fusion of glutamate vesicles. But this spontaneous fusion, this says a lot about the ability, the baseline ability to release glutamate. And this is we call it the release probability. And the higher release probability to release glutamate, the higher the probability also to have an action potential. So meaning this is in fact something which triggers the action potential and the release of glutamate. And this is what we see in fasted animals. This probability to release glutamate is much higher when animals are fasted. And we are able also to block autotaxin. And blocking autotaxin is done by, we call it small inhibitory molecules. So these are molecules which are very similar to the LPC, the lysophosphatidylcholine, choline, but autotaxin is not able to convert them, and it's not, and they have a high affinity, and they simply block the function of autotaxin. And they are very specific. And when we block autotaxin, we see also this increased release probability is going down. And what we have shown in other papers, so this increased release probability is something, it reflects the spontaneous activity of the neuron. So if you have a higher MEPSC frequency, you have a higher spontaneous activity of the neurons. But this is not something we invented, so hundreds of people are showing this by electrophysiology. What we show, what was very fascinating for us, was that these lipids are increasing the release probability and therefore by the spontaneous activity of the neurons. Now, then we said, okay, now we know if animals fast, they have a higher cortical activity, and in fact, this is what we are also thought we try to understand, and this is what I talked at the beginning. So when we have animals which are fasting, we think that they have to search for food. I mean, if there is no mechanism that animals search for food while they are fasting and have no food around, then animals would die. I mean, mice are very dependent on food intake. They have a very high metabolism. I think 3 times higher than we have. They are, after 12 hours, they already depleted their glycogen stores. And 48 hours fasting for mice, it's incredibly long. We say that already 18 hours of fasting corresponds to 2-3 days of fasting in humans. So you have to think that the mice is like a feed forward mechanism, what you see in humans much later. So we have everything much more packed. And then what we know exactly that after fasting, people tend to eat more. Yeah, so I think a lot of people who are trying a diet experience this. When they stop the diet, they have the tendency to eat more during time and especially in the first time after fasting. And we call it hyperphagia. And in mice, we know it very nice. It's called fasting-induced hyperphagia. And you can see in the figure 2, that figure 2b, that during fasting conditions, we tested this inhibitor of autotaxin and there was no effect. But if we are inhibiting autotaxin after fasting, we indeed see that animals eat less. So meaning indeed, this lipid inhibitor is affecting food intake. Now, this is first a general observation, and we need more specificity because we really want to know, is this dependent on the brain? It could be also dependent on peripheral autotaxin, because autotaxin has a big role in the adipose tissue. So it could be that it's simply a far effect where you don't know, an epi effect where we don't know that it's specific for the brain. It may be induced by something else. And therefore, we deleted autotaxin specifically by using a genetic mouse in the cortex. So we took floxed autotaxin mouse, a mouse which expressed floxed autotaxin, and we took EMX1 Cree, which is deleting only in the brain. So you have a Cree expression in the brain, and autotaxin is only deleted in the brain. And then indeed we saw that after deleting autotaxin in the brain, animals eat less. Now we wanted to be even more sure, and we also deleted the LPA2 receptor, because the LPA2 receptor is in fact the effector of LPA at the presynaptic terminal. Now, indeed, we analyzed this mice and saw that after fasting, this fasting-induced hyperphagia was decreased. So indeed, we could show that by blocking lipids or blocking lipid signaling, we are able to decrease this fasting-induced hyperphagia. Okay, now we were interested to see, is this a unidirectional process? So meaning, does it go in both directions? Is it only decreasing this fasting-induced hyperphagia? Or if we increase the lipids, we are able to even top this fasting-induced hyperphagia. And we have animals where we have deleted the PRG1 molecule. And as you look here in figure 2e, after fasting, these animals, which are heterozygous for PRG1, so it's not completely deleted, you have only a gene dose effect, then they are eating more. And why didn't we take the homozygous knockouts for PRG1? This is very simple. We found a human mutation, it's called PRG1R346T, and at the position 346 in the PRG1, this is in the intracellular C-term, there's a mutation at the arginine, which is then converted to threonine. And this is something which is very cool because there is a serine there and you have the phosphatases. It may have something to do with phosphatases, but we had a very long paper on that. It's Vogt et al. In EMBO Molecular Medicine 2016, where we have shown that in fact this mutation is changing the intracellular glycosylation, something we were not prepared for, because glycosylation is mostly extracellular, but there are also some intracellular glycosylation motifs. And this is inhibiting the take-up of LPA at the synapse via PRG1. So this is a loss of function mutation. And this mutation occurs only in the heterozygous, in a heterozygous way. So we analyzed a lot of human cohorts in this EMBO paper, and we were not able to find homozygous mutations for this SNP in humans. We only found heterozygous. And I think we analyzed some more than a few 10,000 human subjects, so we never found a homozygous mutation. So that was very strange. We would have expected it, but it seems that this mutation is not very helpful in a homozygous prevalence, so only heterozygous. And therefore, we only took PRG1 heterozygous animals. But we have created a mouse line which expresses this mutation, and the neurons in these mouse lines are more hyperexcitable. So you see in figure 2G that the MEPSC frequency, so the mini frequency, is significantly increased. And when we let this animal having food after fasting, we saw that indeed this fasting-induced hyperphagia is increased. This is figure 2H. Okay. Are there questions, or shall I continue?
Oh, apologies, I was just listening there. Please
Oh,
continue. Please continue.
okay, good, good. Okay, now one of the questions which then arose was whether indeed this antitoxin is able also to change food intake and the frequency of food intake. How can it modulate this? So I mean, yes, we saw fasting-induced hyperphagia. That's very nice, but it's a very special condition. It applies to people who have a diet, but would we be able to turn around the feeding behavior of an animal? Because that would say, okay, we are able also to induce a long-term effect, not a very specific chronic effect. Because I mean, whenever you have this very strong chronic effect, you think you don't know if this is really is also applying to a long-term effect. And then we analyzed this human mutation, animals expressing this human mutation, and you see it in figure 1C. If you simply weight them, or in figure 1B, if you take animals which are 17 weeks old, the body weight of the PRG1 mutation carriers is higher. And then we said, okay, we start at 17 weeks of age. Maybe this could be some kind of, yeah, it can happen. Yeah, I mean, yes, maybe by chance. We simply follow up with animals. And then you see in figure 1C, so we normalize the body weight at 17 weeks, and then we looked on how this continues. And then indeed we saw that the human mutation carrier mice are gaining more weight than the wild type litters. Then we said, okay, let's see what happens when we take high-fat diet. So not standard diet for mice, but we take high-fat diet. And this is very interesting because this is in fact the Western diet which we are using. So it has a similar caloric energy, but it has a much higher percentage of fat. And what we then took, we took the PRG1 knockout animals, which have a higher excitability. And we wanted to see if this higher excitability also translates during high-fat diet into an increased body weight. And indeed, you see it in figure 3D that the animals, the PRG1 knockouts shown in red, have a higher body weight compared to their wild-type litters. Now, we wanted to go the other way around and say, okay, if they have a defect at the synapse and there we have autotaxin, which is continuously producing LPA from LPC. Then if we block autotaxin, the effect in this PRG1 knockout mice should be higher than in wild types because they already have a higher cortical excitability and they have more LPA around. And indeed, you see it when you give autotaxin to these animals, the decrease of body weight in the animals shown in red is much higher than in the animals shown in dark gray. And these are the wild types. So dark gray are the wild types which received an autotaxin inhibitor, and dark red are the knockouts which also received an autotaxin inhibitor. But these animals lost much more weight than the wild types. But if we would not give an autotaxin inhibitor and only the vehicle in where we have solved the autotaxin inhibitor, and this is DMSO, you still see that there's a very significant change in body weight compared to the non-treated wild types. Now, the question was, how can this come for? How can we achieve this? And we had a look on how animals eat and how much do they eat per day. And you see this in figure 3F. There is the food intake per day during 8 days. And you see when you sum up the total or you compare the total period of analysis, the treated animals eat much less than the vehicle treated animals. Now, as I said before, we found a PRG1 loss of function mutation. This is the mutation PRG1 R346T, only in heterozygous animals. And we tried the same in PRG1 heterozygous animals because we have shown this is a valid model also for the synaptic mutation. And then you see when you treat these animals being on high-fat diet, the animals or PRG1 heterozygous, they lose much more weight than the wild types. And here you see very clear that it is caused by a lower food intake, meaning we have a chronic effect which really depends on the excitability of the cortical neurons. Okay, so that was one part of the story, and possible we will just go to the supplement because there we explain some things. Yeah. So first, you see the extended data figure 1. Do you have this figure? Are you able to see it?
Yeah, it should be all the way on the end
Yep.
of the file. Yeah, thank you.
Yeah, so these are a little bit complicated, very, very beautiful pictures, but they are not self-explanatory. So if you are very deep in neuroscience, it's quite, yeah, we have some understanding of it, but even that is not so easy. So first we look on the autotaxin expression in extended data figure 1. And you see that it's mostly expressed, so the redder, the more red the signal, the stronger the expression is very strongly expressed in the cortex, and especially in the upper layer. And also something in the layer 4 we have, but not in layer 3 so much, not in layer 5. We have it very much, you see this reddish structure in the middle. This is the ependyma, and we know that in the ependyma where you are producing, where the liquor is produced, the CSF is produced, we have strong autotaxin expression. And therefore, indeed, this is a further proof that when we are analyzing CSF, we have a very good impression on what happened at the synapse, because there we have a very strong expression. Other strong expression is in the CA1 region. Now, in the figure B, C, D, and E, we have a stronger expression, and we see where PRG1 is expressed, and you see that PRG1 has a similar expression pattern like autotaxin. When you look in the visual cortex, you see it in layer 2. You see some sparkles in layer 3, but very strong in layer 4, very few in layer 5, and then some sparkles in layer 6. So there's a similar expression like in the in the cortex, and if you look in the S1BF, the somatosensory barrel field, it's in figure F, then you really see indeed red is the expression of PRG1, green is the NOI-N expression, and indeed you see strong expression layer 2, almost very few expression, very few spots in layer 3, but very strong in layer 5, which is very close to the border of a very strong expression layer 4, but very close to the border of layer 5. So this could be also 5A. So it's something which is still on research. And then some sparkles in layer 6. And you see it's not expressed in inhibitory neurons. These are GAD67 neurons, and then you have some kind of interneuronal classes, the parvalbumin, PV, the calretinine, CR, and the calbindin, CB. You don't have it in the interneurons, but in the CB, you have some kind of excitatory neurons which express CB, and they also express PH1. Now, when we look, when we asked why do we look at 18.1 and not on other LPAs, we tested whether in the neuronal setting, the LPA 18.1 is indeed a very strong activator of neurons. And if you look in extended data figure 2b, you see here the effects of 18.1 application. So when we apply 18.1 to neurons, we have an increase in MEPSCs, and this is significant. But if we apply 18.0, we have almost no increase. So indeed, this shows that we are looking for the right lipid. Now, we also tested after fasting the novel object interaction. And this is something that's very interesting because the novel object interaction is a behavioral test which is related to the cortex. It's not related to the hypothalamus, because you could argue if you want, and reviewers tend to argue like this. They say, yes, you are testing something very interesting, but it may be that everything you are testing is on the level of the hypothalamus, because the hypothalamus drives the food intake. Now we wanted to take a non-food object and simply test the curiosity of the mice. This is what we call explorative behavior. And we give the mice a novel object which it has not encountered before. And you look how long does it interact. And you see in figure 3b, extended data 3b, that indeed after fasting there's a higher increase in interaction, and you can completely block this by blocking autotaxin with a substance called PF8380. So this is very specific. And we did all the tests we have I mentioned before. So when you take out autotaxin in the cortex, this novel object interaction is decreased. This is in figure 3c. Or you take out LPA, the LPA2 receptor, also this interaction is decreased. And then we tested vice versa. What happens if you fast animals which have no autotaxin in the cortex? How is the exploration time? And you see it in figure 3D. There's no difference. And then we looked in these animals which have no autotaxin in the cortex and blocked autotaxin pharmacology using pharmacology, because somebody could say, okay, it's a hypothalamic autotaxin which plays a role. No, if you block autotaxin by pharmacology, you have no effect when autotaxin was taken out from the brain or from the cortex. So indeed, this really fits with that. So we went up and down and had exactly these responses which were very clear. And now I go back to the last figure, the figure 4, where we wanted to understand how everything happens. And we have these agouti-related proteins which are expressed in the hypothalamus, and they are characterizing a population. And these agouti-related proteins were shown 2 years ago that they are affecting lipolysis. And this was shown in a model where animals were fasted. So it's a model for anorexia nervosa. Animals get only limited amount of food, but get full access to a running wheel. And animals which have limited access to food or only a very short period where they are able to eat, they develop a hyperactivity and they lose weight. And this is a model for anorexia nervosa. Now, they have shown that these agouti-related proteins are important for lipolysis, and lipolysis is important for animals to survive. So if you block agouti-related proteins, animals which are supposed to this model of anorexia nervosa are dying within days. So this is something which has a very strong effect. But these animals are able to survive if they get fat food, yeah, because you supply the fat which would be released by lipolysis by giving it orally. Now we took these mice and we fasted them and found indeed that LPA was decreased in the plasma and also LPA 18-1. And this indeed shows that these animals are not able to go into lipolysis. And then we looked in the CSF, and indeed we see that the LPA was not really changed, but the total LPA was increased. So that was a very interesting observation. But what we found was that when you look at LPCs in the plasma, and this, if you are able to go to the paper, is in extended data figure 6. The LPC in the plasma, the total LPC, and the LPC 18-1 are decreased. And also other levels are also decreased. 18-0 was decreased and 18-3 was decreased. But what we saw there in the figure 4, I just go back, was that the total LPAs were increased. And what happens when we have more LPA? Then we have a competition between different LPA species for the LPA2 receptors. So then we have LPAs which are putatively not active, but they are inducing a competition for the LPA2 receptor. And therefore, this is known from other people and other analyses, and therefore we calculated a ratio. And when you calculate the LPA 18-0 ratio to the total LPA, you see that the ratio was very significantly decreased in this agouti-related protein deficient animals. So indeed, it's not the absolute level, but it's also the ratio to the other LPAs, which is important. Now we wanted to see if it's true, and we have tested the prefrontal cortex, we have tested excitability, and found that in the prefrontal cortex, there's a decreased excitability in these animals which do not have the agouti-related protein. And if you look at figure 4i, then you see that the food consumption after fasting was significantly decreased. Now I come to the conclusion of this paper. So indeed, if you look at figure 4g, so we have depicted there everything. You see this synapse. This is the cartoon of the synapse, which contains everything we discussed before. So the astrocytes, which are producing the autotaxin, the LPC, which is converted by autotaxin to LPA, and the LPA in the synaptic cleft, which is either scavenged or captured by PRG1 and degraded, or is able to activate LPA2. So this is a balance between degradation and activation. And you have the things which happen much before, when the lipolysis starts. The agouti-related proteins, which are dominating the lipolysis and are producing LPC. But LPC is not able to simply enter the brain. It has a specific transporter. And now this is very cool, this transporter. So this transporter is in fact the bottleneck to the brain. So LPC is the driving force for LPA production. And LPC has to be transported actively via a sodium cotransport with a sodium cotransport. So it's something which is somehow consuming, and it's very selective. And it's not easy to find a lipid transporter. And people who have knocked out this LPC transporter, they found tremendous changed brains, so microcephaly. The animals were small, they had different defects, but really a lot of defects in the brain, so microcephaly. And they found also mutation in this transporter, LPC transporter, in humans, and they had exactly the same phenotype with microcephaly and different phenotypes. So that is the big picture, and this shows we have here regulation of different levels. So when we decrease lipolysis, what happened, why the total APA was increased in the CSF is that there is an increase in this LPC transporter, which we have shown. And then you have a selective transport towards other lipid classes, which are then competing the LPA-18-1 out from the LPA2 receptor, which we have shown in figure 4e. So we have shown that the ratio was significantly increased, and thereby this excitation is going down, and animals eat less. So this is a quite big story, and it's quite complicated, but this cartoon is showing very clear, I think, what happens in the brain. And now I'm happy also to take some questions.
Wow. Thank you so much, Johannes, for that wonderful presentation. I had a question earlier, but my internet was kind of acting up. But
Oh, I'm
um,
sorry, but
it's
please.
okay. And I'm pretty sure there are people in the audience as well that might have questions. So if you have any questions, please come up. Um, or you can post your questions in the chat and we'll read them out. But yeah, before we just, go on, I have a question for you. Could you please go over the mechanism of anorexia that you mentioned? Because I'm trying to imagine it happening in humans, and I just wanted to maybe if you could go through it one more time.
Yes. So this mechanism is a behavioral mechanism. Animals are allowed only a few hours to eat, but they have continuously access to a running wheel. And what animals do, they are running very much, and they have a hyperactivity while having less amounts of food, and they decrease their body weight, they lose fat, and they go into lipolysis and can survive quite well. But if you knock out the agouti-related protein neurons, and this you can do with diphtheria toxin receptors, which they express, and we have agouti-related Cree. So it's a quite complicated system, but it's very nicely genetically. These animals are not able to go into lipolysis, and they die because they are not able to supply lipids while they are in this anorexia phenotype. And that was something that was very interesting because it's shown that indeed this for people who are, or at least for this model, anorexia model, lipolysis is very important, and it's driven by the hypothalamus, which also drives food intake. So that is something that's very nice and links also lipolysis and food intake and hyperactivity and a model for anorexia. Is this answering your question, Cicera?
Yes, definitely. And I was able to kind of just imagine it, kind of in a human model. And I'm hoping maybe this could add to and, propel the research forward that, research on food, eating disorders and, things like that.
Yeah,
So, yeah.
the interesting thing is that there are some papers which show people coming with anorexia for treatment in the hospital, and they really have decreased LPCs. Yeah, and even after the first feeding, they still have decreased LPCs. After gaining weight, this normalizes, and but this initial finding is very interesting because you would think that if this is a continuous situation, you would have an adaptation of the system, but it seems that the system cannot adapt completely. Yeah, so you still have increased or changes in the lipid composition.
Wow, that's very fascinating. Thank you so much for answering that question. All right, so we're gonna go next. Jamie, do you have a question? Jamie, are you there? All right, Katerina.
Hey, thank you so much, Johannes, for this amazing presentation and for this amazing work. I think it's so important that you
Thank you,
discovered
thank you.
the
Yeah,
mechanism. It will probably save a lot of people long-term chronic disease and lives. Improve life of a lot of people, and especially in developed countries. So yeah, thank you for this work. Um, so I have a question about. It's really interesting the glia involvement in the circuitry, especially in the hindsight that the neuron to glia ratio in the human brain is even higher. So that the there are more glia and astrocytes in
yes.
humans.
Yeah,
The
yeah,
high
indeed.
amount. So it's
Yeah,
really
it was a
interesting to
reviewer question. It was a reviewer question. So they said, indeed, you have much more neurons per space and less glia in the mice, and you have more glia and less neuron. The ratio is different in humans. And then we said, okay, we look for the expression, and we look how does it look in humans. And I think we have a picture somewhere. No, I think that was, we only showed to the appreciation of the reviewers that indeed in the human brain, in the cortex, we have similar conditions. So also autotaxin is expressed by glia. You have the LPA2 receptor expressed in excitatory terminals, and you have the PLG1 in postsynaptic density. So, but sure, you're right. It may be that the effects are different. I completely agree with you. And maybe the effects are stronger or even less strong. But what we know from people coming with anorexia to the hospital for treatment, they indeed have this lipid changes, and the adaptation is quite minimal. Yeah, and especially LPC, where you would think LPC it's so abundant in the blood, it could be easily replaced and rebalanced by a lot of mechanisms, yeah, where you produce LPC, LCAT enzymes. But it's not. It seems to be that it's not so easy as we think. Yeah.
Yeah, I would assume that maybe even stronger the effect in humans. And do you think, or you're thinking about looking if there this change? Do you think it's driven by epigenetic changes that it persists, or yeah,
No.
such a strong effect?
Yeah, whenever you talk about epigenetics, I'm sure there is something. Sure, I mean, you imagine, I mean, the production of autotaxin, the production of this transporter of LPC transporter, the lipolysis enzymes, they are very strongly, I guess, they are very strongly regulated by epigenetic changes. But I mean, we take it slow. We just uncovered a non-neuronal pathway, so they are not. They are not neurons which are involved in the periphery. It's LPC, and a lot of things will have to come. I mean, we are just starting the path.
Yeah, it's a very exciting one. Thank you. I want to give Joyce and Dr. Shah and Jamie an opportunity to ask questions. Please go ahead.
Thank you.
Hi. I missed part of it, so I apologize if you covered this at all, but I'm wondering if you have any thoughts on the role of inflammation and in particular related to the microbiota and microbiota changes. Thanks.
Oh God, yes. This is really something very, very cool question. Yeah, it's much further than we are thinking now here. Indeed, inflammation is one of the major driver of brain aging. Inflammation is one of the really important factors which also goes align with higher excitability in the aged brain. I mean, I think you are talking about this, and also depression. There seems to be some kind of inflammation. And yes, I think inflammation plays here a role and lipids play a role, but we didn't touch this in this paper. So this is something we have to do a lot of aging research, and we have shown that these lipids are important for hyperexcitability, which continues or is increasing during in the aging brain. But I'm sure you have a very good lead on that. Inflammation is one of the major driver of brain aging.
Thank you.
Thank you so much, Joyce. Dr. Shah.
Yeah, thank you so much, Johannes. I mean, I was a little bit late, but of course I'll be back and listening to the reply. My question from you is about that you just talked about the agouti-related peptide, and I know that there is some of the researchers around the relationship between the plasma level of the agouti-related peptide and the endometrial cancer. So I was just wondering, do you have any further information around that?
Sorry, I'm not on that topic. I would speculate too much. It's beyond my expertise to answer this question, but very interesting question. I mean, we still wondered how agouti-related proteins are able to influence lipolysis. We have some idea because there are not many neurons which leave the brain and follow the path through the organs, and the vagal nerve is one of the most prominent target for this. But I'm sorry, I have not insight on the endometrial cancer, although it's a very interesting topic. Yeah, I really
Yeah,
believe.
actually, it's exactly, I mean, as a positive significant effect reported, so it can be related. That's why I just asked you, because I know that it's an animal model, but however, I was just wondering maybe you have further information. Thank you.
You're welcome. Sorry for not being able to answer the question in deep.
Oh no, here, Science Society, it's okay to say you don't know. Yeah,
But
it's
possibly, if I
totally
can
fine.
answer something, there's still a link. So I know colleagues are working on endometrial cancer, and the LPA2 receptor seems to be one of the important markers for bad prognosis. But I don't think they have any clue on agouti-related proteins. So possibly very far, and it seems to be that lipids are involved, and also in ovarian cancer. Yeah, but it's really too far to speculate.
Exactly. It's related to the hormonal changes, and also they just found that it's lower in obese controlled women also after they're giving the treatment, and correcting the BMI still has the same
Perfect.
impact.
You seem to be an expert. I admire this. I'm only a visitor in this field.
Thank you so much.
Yeah, thank you so much. Jamie, are you back?
Yes, I am. Sorry about that before. I was looking some stuff up as I was listening to your talk, Doctor. That is an incredible talk. Absolutely amazing. I have to admit, there's a lot of it that I'm very foggy on. So please forgive me when I make any errors in my questions to you.
No, there are no errors, please.
Yeah. The one that was sticking in my mind was when you were mentioning about this mutation in the mice. Is this mutation the same type of mutation that did you say you found this in humans as well? Is this the same mutation? Is it identical?
Yes. So the mutation, the PRG1 R346T, yeah, it's the same mutation as in mice. The homologous mutation is the R345T. I mean, it's one amino acid switch, but it's exactly the same. And what we did, we have analyzed human mutation carriers, and we did PPI, which is in fact something. It's a pre-pulse inhibition, and in in humans, you are testing the P50 wave after double-click auditory paradigm. In the mice, you also have a double-click auditory paradigm, and you test the startle response of the mice, and this is something similar. And then indeed we have the decrease of PPI, which we call an endophenotype for schizophrenia. So meaning this is the inability to suppress redundant information. So you have 2 clicks and the animal reacts at a stronger way to the second click in the startle response when compared to wild type. So meaning it's not able to suppress the redundancy. So the filter function of the brain is reduced. And this is what you know from people with schizophrenia. But we are talking on an endophenotype. And endophenotype is very difficult to define, because a lot of psychiatrists say that it's difficult. What is an endophenotype? And there they say, yes, it's an intermittent or phenotype. So it's in between schizophrenic patients and patients who have not been diagnosed for schizophrenia. But this also means it's one part of the story, and we cannot tell how big the part of the story is, because we know that in schizophrenia we always have a multifactorial disease. So there are a lot of very fancy hypotheses where you say the 2-hit hypothesis. So you have to have a predisposition, and then you have to have a bad event, which is then starting the disease. I mean, these are very nice explanations, and I think you find retrospectively a lot of confirmation for this. Yeah. But what I want to say is this human mutation is an endophenotype. It's a predisposition. And what we found here with the fasting, it seems not to be very specific for schizophrenia. It's also even more important for food intake. Yeah? Yeah, but that's another story.
That definitely does fascinate, that they're both connected, and that it's connected in such a way. Yeah. I'll let someone else on the stage ask a question if they have any, and if there's time, I'll come back with another one for you. Thank you very much, Doctor.
Thank you, Jamie, for the questions.
Yeah, thank you so much, Jamie. Does anyone else on the stage have another question? Or in the audience?
Well, I have a question regarding, are you planning on maybe doing clinical studies next? First with ATX related drugs, and then modulators, and then also I could imagine, do you think there's a vulnerability through environmental insults in these different levels that, you know, could lead to like obesity and diabetes type 2 driven by this mechanism, and that some sort of, stress factors kind of also increase this level, and not just
Sure,
dietary
sure.
changes?
Yeah, I mean, we have here a multifactorial metabolic disorder, and we are just tackling one part which we can manipulate genetically. And environmental stress is sure a part of the story. We also tested resilience in this mice, in the PHG1 human mutation mice and this PHG1 deficient mice, and they have a lower resilience. But you don't see it immediately. You see it when you first apply a stress on the animals. So you first have to stress the animals, and then this resilience deficits, which you can measure in different behavior strategies, then that comes up. Yeah. So indeed, what you say, it seems to be I really can imagine that environmental stress is a contributing to this disorders. And if you have a prevalence, sure, this may drive it in one or the other directions. But I'm here. I have no idea on how it will work. So what we tested was food intake. What I can also imagine experimentally to test food intake and stress, because you can apply stress protocols for animals, and then see how this decreased resilience applies to food intake. This is something which is quite interesting. Yeah, so for sure. But I think the difficulty on your question is not that the question is not possible to answer, but it's difficulty to design an experiment where you exclude other causes. So you want to have it most you have to have a most clear answer, and this is not always possible. You can control a lot, but not everything.
Yeah, especially in humans, I agree. We had yesterday a guest speaker here. He was talking mostly about Alzheimer's and brain imaging, but then he also went into PTSD, and I had then the idea, uh, you know, for vulnerability to use firefighters as a group instead of soldiers, because we hopefully don't want people to go into war, but firefighters have a very high occurrence of people.
Yes. Yeah.
So maybe when people apply to become a firefighter to take some samples, I don't know how easy it
Yeah,
will.
yeah, yeah. I mean that for sure. PPI would be a stressor, would be something which you have to test if it's applicable, but would be an endophenotype. You have to test them and make neuropsychological tests. We for example know that this PRG1 deficient mice have a higher motoric. They are hyperlocomotion. They have a show a hyperlocomotion. They have a better motoric performance. So indeed, it's not so far away of thinking that. Yeah, it's feasible.
That's so interesting. Did you think of using
Oh, 1 second. Our room cleaner just went on. Yeah,
oh,
sorry.
don't worry. Um, did you it would you think it's interesting to look also into ADHD type of models, since you have this
Sure.
hyperexcitability? Maybe it's also related to this pathway. I don't know.
Yeah, I mean,
That's
this is the hyperactivity disorders are quite complicated. I mean, sure, yeah, there are different models. Shank-free is one of the models, and they are, I think, 4 different mouse lines. They show exactly opposite behavior. Sure, would be very nice. Yeah? And I think what we have shown here, this may be an accelerator of the phenotype in some mice. Yeah. Because it's an endophenotype. It's something which brings up other phenotypes which are mostly hidden. Yeah, this could be, but that's it's. We didn't start with this. Yeah, it's a lot of work. Yeah.
yes, of course. Yeah, I understand. I was just thinking it would be quite amazing if, the drugs. I think you're starting to design a human drug that targets the ATX inhibitors. Yeah.
Yes, we are starting this, but this is a long way. But I like your idea very much with firefighters. I mean, we should test them for this human mutation, and my bet is that they don't have
Yeah.
it. We tested a lot of humans, and yeah, it's not. You have to look very close, but stress is not the best thing. Yeah, I mean, for nobody really is. Yeah, stress a good thing. If you, at least if you think it's a positive stress, but firefighter are really stressed. I like this idea very much. Thank you. Yeah.
Oh, great. Yeah. Yeah. When I did the PTSD study, I needed, I wanted to look into more groups, and one of the groups was firefighters, but it was different. It was language detection related study, and it was a very interesting comparison group.
Yeah,
And
it's
a
it's
very
yeah,
high,
yeah,
yeah. So
because they have to be stressed. I didn't thought about this, but you're right. Very very cool idea. Yeah.
yeah, wonderful. We've been going almost an hour and a half. I wanted to check with you. Um, probably. And we probably
Yeah, if
already going over the time you planned for this, so, but we will
yeah, I mean, if there are still a few questions, we can answer the last questions. Otherwise, I have to look if my children are sleeping or they are looking on Netflix in their bed. Yeah,
Oh,
they know when I do not control them, they start doing nasty things.
of
Yeah.
course, all of our kids do that. So
Yeah, but I think it's normal. I
yeah,
mean, we always are a little bit embarrassed, but if you take a little bit distance, you look with a distance, it's a nice thing. I don't think we were very much different. Yeah.
I think it's a sign of, well, that's just my personal theory. If kids don't try, it's a sign of depression almost. You know, when dogs like our different behavioral models, when mice don't even try anymore, to get to like achieve things or try different things. It's a model for depression, right? So if kids don't try to like
Yeah, it's also a view.
change
Yeah.
boundaries, it's probably a bad sign. So that's good. It's good.
I agree with you, Katerina.
Okay.
Definitely. I think Jamie has one last question for Johannes.
I, I do actually, Doctor. Thank you. My question was, I was really interested in what you were talking about with the sodium co-transporter. When you were talking about this, please correct me if I'm not understanding this properly, but when you were saying the sodium co-transporter transports the LPA, that's what's responsible for that, yeah? You said that's the bottleneck with the LPA
Yeah.
getting to
No,
the
it's in fact the LPC. Yeah, the
LPC,
LPC,
sorry.
lysophosphatidylcholine, which is released from the liver, and there are huge amounts in the blood plasma. And this LPC is transported via this transport. It's called MFSD2A. Yeah, so this is a long name. And the LPC is the amount of LPC is the bottleneck for LPA production at the synapse. So that is the point, because uh, even if we have a lot of LPA in the blood plasma, it will not pass the blood-brain barrier. And even if you would have some kind of LPA transporter, which you never know, it could be, yeah. LPA is very unstable because you have it's non-saturated and is rapidly degraded by lipid phosphatases, which are abundant in the brain. So it has to act locally, and it has a very short time of action. So therefore, LPC is the one which drives the LPA production at the synapse, yeah, and the transporter transports LPC.
Actually, that just totally answered my question, because it was going to be what happens if the transporting doesn't work properly. But thank you very much for your time, Doctor.
Okay.
Thank you for answering my questions as well. Thank you.
It was a great pleasure for me to talk to you, and a very great pleasure to answer your question, because I see you come from different specialities, and you have very different background, and this is for me fascinating. And thank you very much for the firefighters. I will watch the films with firefighters now in a different way.
Oh, thank you. Thank you. It was a great pleasure having you. And we wish you all the funding and all the best for your work. It's really important. And yeah, maybe you come back with firefighter updates.
Surely,
In
let's
the
meet in 1 year back. Yeah, from in 1 year from now.
Yeah, that would be wonderful. We would appreciate that. Thank you so much, Johan.
Thank you very much. You're an extraordinary audience. I really enjoyed to discuss with you the paper.
Thank you so much. It was a pleasure hearing your talk. And yes, please come back whenever you have found out more. Thank you so much for your time.
Yeah, we did appreciate you. Thank you so much.
Thank you.
Thank you, Cecilia. Thank you, Katarina. And thank you for organizing this. I know this is, you do all on extra, so this is, I appreciate it a
Oh,
Oh,
lot.
it's
yeah.
always a pleasure.
Thank you. Thank,
Thank you very much.
thank you.
Okay,
Bye, everyone.
Thanks.
bye.
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 Close the room in three,
Bye, everyone. Oh,
two,
bye,
one.
everyone.
Bye, everyone.